lecture about ATP. But in the first half, I want to talk about this. This is a picture we took in Namibia a few years ago. And Namibia is a vast desert area, really spectacular views. But what I found so striking in this picture was that in the middle of all of this dryness and the sand dunes and this desolation, here's this wonderful tree, this single tree, this acacia tree here. And it just illustrates for me what marvelous biological organism plants are. After all, plants can live on almost nothing. All they need is some minerals, some salts in the soil. They need water, and these acacia trees can send roots deep into the groundwater, and they need energy and from the sunlight, and they can make everything else. So I don't think that plants get the respect that they should. Plants are in some ways the most complex of all organisms. So they have as many genes as we do. They have 25,000 genes. If you look at cells in plants, they have all of the complexity with the nuclei and the mitochondria and all the other structures. But they have additional complex structures that we as animals don't have. And we're going to talk today about the chloroplast in the plants. The plants can synthesize all of the amino acids. Plants don't need vitamins. They make all the vitamins. They require no food from other organisms. They can take CO2, water, sunlight, minerals, and make everything else. And that is essential to our survival. So a biochemist summary of what is life is that for animals, we take food, and this is the formula for simple sugar like glucose, and we use oxygen, and we somehow combine these such that the carbon in our food ends up combined with oxygen to produce CO2, and the hydrogens and the other oxygens in the food come off as water molecules. But what drives all of this complex biochemistry? Well, it is ultimately the sun and the ability of plants to capture the energy in the sun. So if we look at the surface of a leaf with a microscope, we find that it is composed of large cells, and they have nuclei and mitochondria and everything else. But they have these prominent green organelles, the chloroplasts, and it's in these chloroplasts that all of this magic happens. All right, so I first got really interested in this when I was in grade school, and I don't know how many of you remember this, but the telephone company, Bell Telephone, had a prominent scientific lab, Bell Labs, Nobel Prize winning lab, and they produced these instructional videos for grade school kids. And one of my all-time favorites was Our Mr. Sun. All right, now Our Mr. Sun explained how the sun radiates all this energy onto the earth and how we use it in our lives. And this was a very well-made movie. In fact, it was produced and directed by Frank Capra, and the stars in it were Eddie Albert and Lionel Baltimore, Frank Baxter. But towards the end of the movie, they were talking about how photons from the sun, rays of light from the sun, hit these chloroplasts. And the narrator said, and by a secret unknown to man, some little genius inside the chloroplast would grab a snatch of sunlight and a little bit of water and some salts and would make food for all of mankind. And after this step here, this little guy pulls this curtain across so that you can't see how he's assembling all these things inside the chloroplast. And I remember thinking to myself, when I was like 7th or 8th grade, I wonder if they will ever figure that out. Well, we've figured it out in quite a bit of detail, and that's what I'm going to tell you about today. Okay, so Where this magic happens, where we get our energy from, is sunlight hitting the chloroplasts and specifically interacting with the proteins that are in these stacks of membranes inside the chloroplasts. So the chloroplast is a very complex structure. We can just see it in a sort of cartoon. It has an outer membrane here. Then it has these little kind of little flattened spheres inside with a lot of extra membrane. And the really important reactions occur on the membranes of these compartments inside. And the fact that there's a closed compartment here, there's an inside to it and an outside, turns out to be a key to explaining how we make molecules that are the source of energy for all living creatures, the exception of a few things that live in hydrothermal vents, etc. All right, so early on, it was when people were studying how light somehow is used by plants, they learned early on that plants emit oxygen, all right, and they could use this emission of oxygen to find out what kind of light was absorbed by the plant. So this was a really clever experiment. They took an algal cell here that forms a long filament. And this cell had lots of little chloroplasts inside it. And they put it in a tray of water with some little bacteria swimming around in it. And these bacteria were the kind that need oxygen to live. And so the bacteria would cluster themselves in the areas next to the algae where oxygen was being produced. So what they did then was to simply use a prism to shine a spectrum of light. So on one part, they were shining blue light, and in the middle, they were shining green light, yellow light, red light. And what they found is that the bacteria clustered around this area here where there was this reddish, yellowish, reddish light, meaning that there was oxygen being produced here, photosynthesis was happening. The green area was relatively devoid of bacteria, so apparently no photosynthesis was being driven by green light. And then the blue light was very active too. And what this seemed to show is that if you looked at the wavelength of light, it was bluish light and reddish light that drives photosynthesis. And that's, of course, why plants are green. They absorb the red light, they absorb the blue light, but they don't absorb the green light. The green light is reflected or passed through, and that's why we see plants as green, because they don't use that light. Now, we now know that, well, what is really absorbing this light? And it is done by pigments, by colored molecules inside the chloroplast. And the major ones, I'm sure you've heard of, are the chlorophylls. And chlorophylls will absorb light in the reddish part of the spectrum, over here, the longer wavelengths, and in the bluish part of the spectrum. And there are a number of other pigments too, but you see they're very ineffective in absorbing in the green part of the spectrum. So let's just take a little bit of a closer look at a chlorophyll molecule, because what we're going to talk about on this slide is the key to the whole thing. When we talk about organisms having energy, it all comes from this first initial step. So what happens is that a ray of light, which is a photon that's in blue light or in red light, that ray of light goes through here and hits this molecule, chlorophyll. And the chlorophyll molecule is special because it has a whole ring of electrons that are shared over this structure. So every junction here is a carbon atom, carbon, 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 carbon. And you have what is basically a cloud of electrons. And what happens is when a photon comes in here and hits one of these electrons in this cloud, the energy in the photon is absorbed by the electron, and the electron becomes more excited and moves to a higher level. Now, let me just say a little bit about this because it's so central. You know, it would really be important to really understand this if we knew what a photon was and we know what an electron is. And I've been teaching this stuff for many years and have really tried to really understand it, but I'm afraid I can't tell you what they really are. I think when we think about it, we think about little balls spinning around the nucleus, but the physicist and the chemist would say, you know, that's wrong. That's very inaccurate because photons and electrons live in this world of quantum mechanics, and they have this wave particle duality. They behave like waves of energy, and yet in other circumstances, and this is a perfect example, they behave like a particle in that a single photon can hit a single electron in chlorophyll, and they interact, and that energy in the photon can be absorbed by the electron. So I can't understand and can't explain to you the quantum mechanical explanation of what photons and electrons really are. I think you just have to be embedded in the mathematics to sort of see it, but even there, I find that the human brain is just not structured to have a view of the quantum mechanical world. But this old thing that we were taught in school about the ball circling around the nucleus is a useful analogy. We just have to remember it's an analogy. And so I'm belaboring this a little bit, but basically you have an electron in this chlorophyll atom that's circling around at a sort of medium level energy level, and then it gets hit by a photon, and then it goes to a higher energy level. And everything in life depends on the energy that was absorbed by that electron from that photon. And the fate of that electron is losing that energy, going down to lower and lower energy orbitals within different atoms as it gets passed from one molecule to the next. And we're going to explore just a little bit of that. But really, that's the heart of it. Photon hits an electron, it gets excited, and then that excited electron and the energy in it is used to drive all of life. Now, there's one circumstance in which you can actually kind of see this. So here's an experiment that I used to do in the labs. I mean, when I was talking to live students, etc. It's a really kind of beautiful illustration of this. So what is in this flask is some chlorophyll. So there's a little bit of an organic solvent in here. Chlorophyll is actually kind of fatty. It's not very soluble in water per se. And you can see that because if you mush up spinach, the water stays clear for the most part. So you have to add something like a little acetone or something to partly dissolve the chlorophyll. And what you see is green light. And that's because the light that goes through here is all the colors of the spectrum, but the green doesn't get absorbed and we see it as green. However, if you put it in the dark and you shine white light on it, then some of the photons here are going to be in the red region or the blue region, and they will be absorbed and excited in these chlorophyll molecules. But they are unstable in that state. And so after hopping up to a higher energy level, then they settle back down again, and the energy that they absorbed is given off in a couple of forms. And in one way, they give it off by re-emitting a photon. And generally, they will absorb in the blue, for example, and then they will emit photons that are red. And you can actually see them. So you shine white light on chlorophyll, and it will glow red because it's absorbing the light and then re-emitting it as red photons. Okay. Now, before I'm going to, let me just questions at this point, because now we're going to go into the biochemistry of that. But any questions on that sort of basic physics of what happens? Okay, I'm not hearing it, so good. Okay. All right. So going back to our Mr. Sun and whether scientists would ever really figure out how that worked, well, here's what we know. So the chlorophyll inside the chloroplast is held in a particular orientation to maximize absorbance of the light. So it really forms like an antenna, and it contains many chlorophyll molecules and many proteins. Let me just go ahead and show you that we now know the detailed structure of this light absorbing antenna in the chloroplast. And so what we have here is everything in green is a chlorophyll molecule, and they are held in a certain structural array by proteins. That's what the gray helices here, these are proteins that form a binding site for the chlorophyll. And then right in the middle of this whole cluster of this antenna is sort of a central receiving spot. So let me go back here, this sort of cartoon version. So every one of these green spheres here is one of these antenna complexes with hundreds of chlorophyll molecules. So what happens is a red photon comes in, hits a chlorophyll in this one, it passes, and what that happens to that chlorophyll is it passes it on to its neighbors. It almost behaves like an electrical wire, and that high energy electron eventually ends up in that special chlorophyll molecule that's right in the center of this antenna. So if we go back to this one, this chlorophyll right here might have been hit by a photon. It'll get passed on till eventually it ends up in the center, and it ends up right here. All right. So what I'm going to do now is you kind of walk you through step by step of how we get the energy in this photon and put it into some molecule that's stable and that we can use to make all of our proteins and DNA in our membranes, and that we can use it also to provide energy for things like walking across the room or degrading our food. These things require chemical energy, and this high energy electron is going to be the source of that chemical energy. Okay. So the other thing is, this is going to provide you some information that's relevant to climate change also. I'll talk about the tie-in a little bit here. So what we talked about before was photon hits a chlorophyll, generates a high energy electron. And as I showed you where that chlorophyll was fluorescing red, you've got to capture this electron and put it in some safe, stable molecule quickly. And this is something that occurs in the order of picoseconds, less than a billionth of a second, inside a large protein complex that sits in a membrane in the chloroplast. So step one is this excited electron from chlorophyll gets transferred to a molecule called quinone. Quinone is rather simple. You see a little structure of it up here, but it's a similar story to chlorophyll in that once again, you have a situation where there's a ring of chemical bonds that can kind of share electrons, and by sort of sharing the high energy that's in this excited electron, it will stay located with the molecule rather than going flying off as a photon. So step one, the excited electron goes to a quinone. So you end up with this electron now over here on the quinone. Now, the other way you can stabilize this is that this is an electrically charged molecule now. It has this strong negative charge on it. And so out in the water here, we have water that's constantly dissociating, forming OH minus and H plus. And these H pluses will avidly bind to this E minus. And so what you end up with is a quinone that picks up an H plus. It's got the excited electron, and now it is in the form of QH. So it's still carrying our high energy electron, but it's in a rather more stable form. It's not going to fall apart and re-emit this electron because we need to use this electron to make food and energy. All right, so what happens in the next step? Well, meanwhile, back at the ranch, this chlorophyll has lost an electron. It is strongly positively charged, and it wants to replace that electron that it had passed on to the Q. Where does it get the electron from? It gets it from water. So there is a binding site here where a water molecule will bind, and the chlorophyll that's missing an electron has such a strong affinity for electron, it'll strip it off the water molecule. And what's left over? An oxygen and two hydrogens that are missing electrons. This will occur in a couple of steps. So every time an electron gets transferred over here, it'll take an electron off of one of the hydrogens, and then it'll take it off the second electron hydrogen. So you end up with this. This is important to life because this is where oxygen comes from. So it's an interesting way the whole life system is organized, is that oxygen is basically a byproduct, a leftover, if you will, of the chemical reactions that we use to harvest energy from the sun. Now, the other thing that happens is that now you've got these H pluses accumulating inside this compartment. So, key event in photosynthesis is that oxygen and the H pluses, protons, are produced by the splitting of water splitting reaction, catalyzed in part by chlorophyll. All right, but we've got to go back to our high energy electron here and find out how we're going to put it in some form that we can use. So what happens is there's another enzyme that essentially takes the high energy electron and puts it on a really stable carrier. This is basically a protein that contains the molecule NADPH, stands for nicotinamide adenine dinucleotide phosphate, etc. It's a rather complex molecule. Once again, it has a ring structure that can carry the high energy electron in a stable form. And this can be a soluble molecule. It can be released from this protein and float around here in the water, carrying this high energy electron. And we use that to build molecules like DNA and proteins and sugars. And I'll show you one example in a bit. The proton that had been picked up over here is dropped off on the other side of the membrane. And so this transfer not only puts our electron in a stable form, but it contributes additionally to this accumulation of protons that are occurring here on the one side of this biological membrane. So overall, the summary of kind of what happens here. Okay, I accidentally muted myself. Go back here. All right, so overall what happens is the energy and the high energy electrons are poured on this molecule, NADH, oxygen is made, and a proton gradient is generated. And this proton gradient turns out to be essential to understanding where we get energy in our body. Okay, so I'm going to stop briefly just to allow people to ask questions about that part. That's the most kind of biophysical part of what I'm going to talk about. But, you know, it is the fundamental basis of life. So questions, comments from people?
I had a question.
Yeah.
You show you're shining you're shining white light onto the chlorophyll in solution, and you're saying it absorbs blue light and re-emits red light.
Yes.
And that either means that you're doing emitting two photons for each one that you absorb, or else you're taking some of the energy from that blue light, which is higher energy than the red light, and putting it somewhere else. And I was just wondering if you know where that somewhere else is.
Well, part of it will go off as heat. And so, you know, the light will actually, if you shine that on chlorophyll, you can actually measure the heat. Yeah, I thought of that myself while I was putting this together, Mark. But you make the key point that people should know is that the photon emitted is always of a lower energy than the one that was absorbed. So I think there may be an entropy component in there too, but I don't really have the right answer to your question. I know part of it is heat, but it's always, you come out with a net loss.
Some of it's not radiated, it's just heat. It's fairly typical of a lot of fluorescence processes. So.
Yeah.
Okay.
Okay, other questions or comments?
Well, the one that I put in the chat was are chloroplasts descendants of captured bacteria like mitochondria are?
Yeah, I'm glad you asked that, but the answer is yes. Yes, just as mitochondria were free-living bacteria, chloroplasts are also, and they have their own DNA, they have their own ribosomes, and from studying the structure and sequence of this, we can figure out exactly what kind of bacteria they were, but they were early photosynthetic bacteria, and we know which of the classes now are their most likely ancestors. Yep. Okay, any other questions? All right, so let me go back then. Okay. So kind of part two of the energy story. Really, the major source of energy in all cells, the kind of gasoline that runs every living creature, is this molecule ATP. And I may have mentioned before, it's kind of surprising that I think most people have a kind of a rough idea what DNA is. DNA is in the popular literature, but ATP, much less so. And it's really important. A cell can live without DNA, can't reproduce, but your red blood cells, for example, they have no nuclei in them. They have no DNA in them, but they have to have ATP. All right, so let's take a look at just exactly what this molecule is. So that stands for adenosine triphosphate. This is adenosine. It is, in fact, the same A that is in DNA and RNA. When you talk about A, T, C, and G, nucleotides, well, ATP is a nucleotide. So not only is it used to make nucleic acids, but it's used as the major energy source for hundreds of reactions in all creatures. So there are kind of two chemical molecules that have carbons and nitrogens in them. There's a sugar, and then the triphosphate, the yellow here, is a phosphate molecule. So you have phosphate connected to oxygens. All right, and you have a chain of them in ATP. Now, one way to think of ATP, again, it's an analogy, a mechanical analogy. How the energy is really contained in ATP is explained by the language of quantum mechanics, which is a language that that I frankly do not understand. I've tried, but I do not understand it. But an analogy would be when you take a phosphate molecule and you stick it on the end of this, it's like there was a spring there and you have compressed the spring. You've put that phosphate on the end, you kind of latched it to the next phosphate, but the electrons form the energy kind of equivalent to a spring, such that with hit in just the right place, that can be released, the spring will push apart the phosphates and provide energy to drive some process, like moving your cells in your muscles. All right. So what we have is the formation of this ATP by in some way forcing a phosphate onto the end of the chain, compressing the spring in our analogy. That is done in both plants and animals in our own cells inside the mitochondrion, a little organelle descended from bacteria that can do this trick. I'll show you how it's done. And then inside the mitochondria, it is synthesized and then released into the rest of the cell. All right, so you now have the fully charged ATP. It goes out into the rest of the cell and it will bind molecules like those that are making proteins, for example. In previous lectures, we showed how in the ribosome, it attaches one amino acid to the next. The mRNA marches along. Every time you get a new amino acid added to a chain, it requires the energy of a couple of ATPs, or closely related molecule, GTP. All right. And then that means that then the phosphate has come off and you've just got ADP left, and that goes back into the mitochondria. The phosphate has to be put on to make more ATP, which is released into the cytoplasm. So in our bodies, this is occurring all the time. And it's been estimated that the turnover, if we just estimated the total amount of ATP we make each day, it is more than 100 pounds of ATP per day. Now, how do you do that? Well, by recycling it right away. We don't actually use it up. We just take it apart when it gets used to pump our muscles, and then we put it back together again. And what I'm going to do now is look at this amazing process by which this occurs. All right. So when we first looked at chloroplasts, I emphasized the fact that the membranes in which these reactions take place are in a closed compartment, these sort of squashed spherical vesicles. So here's a cartoon version of one of these. In all the chemistry that we talked about before, it is up in the upper half here. So photon of light hits the chlorophyll in this photosystem. That's where water is split to produce oxygen and protons. And then remember that the quinone picks up a proton on one side and ends up dumping it on the inside. And then this excited electron eventually ends up on this molecule, NADP or NADPH. They're two closely related molecules. But let's go back to these protons. So one thing that is generated from this whole process here, ultimately from the energy from the photon, is an accumulation of H plus inside. So it becomes very acidic. H plus is acid, and it becomes very positively charged. Outside here is deficient in H plus. It's got more OH minus, and it's negatively charged. So there's an energy gradient across the membrane. And that gradient is used to take ATP and phosphate and jam them together to make ATP. The person who really figured this first out was an Englishman named Peter Mitchell. And it's a fascinating story because biochemists struggled with this problem for years because they thought, well, making ADP has got to be done by some enzyme that we could isolate from the cell. And Peter Mitchell said, no, the key thing is a proton gradient. Well, what the heck is a proton gradient? It was just a hard concept to explain and to get people to accept, but the experiments in the end show that Peter Mitchell was right, and that's how it works. So the way this, linking this proton gradient to the synthesis of ATP is done is via one of the most amazing enzymes in the cell called the ATP synthase. And I'm going to flip it over from the way it was in the previous cartoon. And here's the basic structure of this molecule. Knocked off my power cord here. All right. Basic structure of this enzyme. So this is a big enzyme that has many polypeptide chains. I'm going to show you a detailed structure in just a little bit. But the way it works is it's basically like a water wheel, or more accurately, like a turbine. So to kind of anthropomorphize, these protons want to go from the inside of that vesicle to the outside. They move because there are many more inside than there are outside. So they'll just flow downhill a gradient from high concentration to low. They'll also move because they're positively charged and they're filled with a place that has many other positive charge, and they're repelled by it, and they're attracted to the negative charge on the other side. So the way it works is they move through the membrane through a particular path made by the proteins, and it is structured such that as they flow through this, they force this whole part of the protein to rotate like a little motor. So here's a nice animation that has been made of what's going on in the membrane. There are certain parts of the protein that contain negatively charged amino acids, and they're sort of arrayed on what forms a wheel in the membrane. So all these protons that have accumulated here can go through this channel formed by a sort of hole in the protein. They can hop onto one of these negative charges, and as everyone hops on, it pushes the wheel a little bit in this counterclockwise direction. And they ride around to the other side until they get to a channel where they can leave, and they have thus crossed the membrane. So this is the key to the whole thing. The force of these H pluses moving through and causing this wheel to turn generates energy in the form of a spinning turbine. And now we can connect this turbine to the synthesis of ATP, and that's in the next step. Because up on the top of our enzyme, let me just go back here. So here was the turbine. Now we're going to look at this shaft and the ATP, ADP binding parts up on top. So here's just another way to model this. We have these proteins, and the red and the green ones are where the binding sites for ADP, phosphate, and ATP are. And the blue and the gray one here are the shaft connected to the turbine down below. So what happens is as the protons force this to spin, up in the upper part of the membrane, you have the lump here on the protein that pushes against the parts that bind ADP and ATP, cause them to swing open, close up again, and then repeat the cycle. And now let's look at this from the side. Okay. So our shaft is here, and it's going to rotate. And right in here is where ADP and phosphate bind. So we're using the energy in the proton gradient to make this spin around. It opens up. ADP and phosphate will bind right there. Then it'll close up. They'll form ATP. Then when it opens again, the ATP comes out and the ADP and phosphate go in. ATP is formed, opens up, ATP leaves, new ingredients go in. And it does this at a rate of 400 per second. Now, in the enzyme world, that's very slow, but as you see, we have a rather complex phenomenon. So this is really bizarre in a way because, you know, with the difficulty of explaining things in quantum mechanical terms, it's so hard to understand how the photoreactions work. But here it's almost like a highly miniaturized mechanical machine, a motor. And this is how it really works. And when this was first proposed, this was felt to be just crazy because, you know, the atomic world doesn't operate like the machines we would build, you know, in a workbench, but this is one that pretty much does. All right, so let me go here. This is about
life requires energy. The universal biological fuel.
Okay, this is a 4-minute movie that really shows the detail of this amazing enzyme in some detail. And you'll be able to see the ATP and the ADP and the whole formation process. This come out of the lab in England, and the woman involved here was one of the key people who first really showed the structure of this amazing enzyme. And so this is kind of where we stand with it today. How do you make this energy molecule ATP?
Is a molecule called ATP, or adenosine triphosphate. ATP stores chemical energy in the form of a high-energy phosphate bond. As the name suggests, it consists of an adenosine ring with a chain of 3 phosphate groups attached. The last of these phosphates can be split off to release the energy within the bond, resulting in adenosine diphosphate and free inorganic phosphate. This energy is needed to keep cells alive and is used from processes from muscle contraction to producing the thoughts your brain is having right now. The hydrolysis of ATP can be reversed by the addition of inorganic phosphate to ADP. However, this of course requires energy and is performed by a molecular power generator called Atp Synthase. Here we see an atomic model of F1-FO ATP synthase sitting in a membrane. This molecular motor is found in the mitochondria of eukaryotic cells and is responsible for making most of the ATP. Mitochondria, the powerhouses of the cells, turn over around 60 kilograms of ATP every day in an adult person. A long chain of biochemical processes convert the energy stored in food into a proton gradient across the inner mitochondrial membrane. These protons drive a rotation of the turbine within ATP synthase, resulting in the synthesis of ATP. Like all generators, ATP synthase consists of 2 separate motors coupled together. Within the membrane, we have the FO motor, named after the binding of antibiotic oligomycin, and at the top we have the F1 motor from factor 1. The FO motor is a proton-powered motor. It is thought that protons flow through a channel open to just the intermembrane space, where they bind to a ring of protein subunits, rotate 360 degrees, and exit through another channel exposed only to the matrix. The net flow of protons driven by the proton motor force provides the energy for the generation of rotation. The torque generated in the FO motor is transferred to the F1 motor by a central stalk or shaft. The F1 motor is responsible for generating the ATP by addition of phosphate to ADP. The top of the central stalk acts similarly to a camshaft, so that as it rotates within the F1 motor, it causes conformational changes of the catalytic subunits. The catalytic unit is made of a dimer of subunits, and there are 3 of these arranged in a ring. Catalysis occurs at the interface between the dimers. If we concentrate on one dimer, we can observe 3 distinct states. First, ADP and phosphate bind to the catalytic side. The central shaft then rotates 120 degrees to rearrange the molecules. Next, the enzyme undergoes a further 120-degree rotation, and the ADP and phosphate are fused together to create ATP. The enzyme then rotates again to return to the starting position, where ADP is released and ADP and phosphate can be bound for the next cycle of catalysis. One key aspect of this relationship is that catalytic subunits must remain stationary with respect to a rotating central shaft. This task is performed by a scaffold on the outside of the complex, referred to as the peripheral stalk. Recent work has shown that what was once thought of as a rigid scaffold is actually dynamic and is able to accommodate the changes necessary for most efficient function. Looking at this machine in its natural environment by electron chromatography of intact mitochondria has shown that ATP synthase dimerizes to shape the mitochondrial inner membrane into their signature cristae shape. This turbocharges ATP synthesis by focusing the proton gradient near ATP synthase.
Okay, so yeah, the little bit at the end there, as though it wasn't complicated enough, then they bring in dimers of this complex enzyme that interact. It's really amazing. So I've been really always interested in this because it turned out that this enzyme, it really turned out to be quite closely related to the work that Rusty and I did in our lab. So when we came here as, young research people, we were looking at the question of how do our protons pumped across the membranes inside the cell. So we knew, and we were involved in the early discovery of enzymes that take ATP and they split it to make ADP and phosphate, and that energy from taking that high energy charged phosphate off the end is somehow used to drive protons across the membrane. Now the reason this happens in cells is because you have these organelles, vacuoles, and lysosomes that form like little stomachs in the cell. And your stomach is an acid place, it's full of acid to help digest your food. Well, these little organelles get filled with acid to help recycle molecules inside. And so we eventually were able to isolate this protein and find the genes that encoded it. And it turned out to be a direct relative, the big sister, if you will, of that enzyme that makes ATP. It runs in the opposite direction. It uses ATP to move protons rather than have protons use ATP. And just in the last year, they've finally come up with a structure. We didn't do this. So this was done by people using the big synchrotron radiation source in Stanford, et cetera. But they now have a complete structure of this vacuolar ATPase, and they are just amazing molecules. The turbine is down here, the central shaft is here. So in this case, ATP comes in, it forces this to rotate the shaft, and then as the shaft rotates, it forces protons to go from this side across the membrane into the inside. So all reactions are in principle reversible, and so the reaction that is used for ATP synthesis in the mitochondria goes in the opposite direction, driven by a different enzyme in membranes like the vacuoles and the lysosomes. Okay, so I'm going to take another opportunity for people to ask questions there, questions about how all of this works. Pretty heavy duty biochemistry, but these central explainer of what energy is in living creatures. Any questions, comments?
I have one. How many protons does it take to make an ATP? How many protons do you have to run through the machine to get an ATP to come out?
it's interesting, a straightforward question that has been the source of decades of controversy and arguments, angry ones sometimes. But the best guess right now, it's not a guess, but it's probably 3. But interestingly, it looks like it's not a simple decimal number. Once we've worked out the detail, you know, the torque in the machine it's not exactly a one-to-one thing. There turn out to be, in some cases, 10 proton binding sites. So to go around is not divided by a simple integral number.
So you do a revolution with 10.
Yeah, exactly.
Proton sites.
And there's some variation in that, in creature to creature, but they all have the same basic structure there. Any other questions? Okay, all right. So let me then go back to kind of part 3. Okay, so we've looked at synthesis of ATP. And as I said, that takes place inside those chloroplasts, but the same process with the same kind of enzyme takes place in our mitochondria. So in the chloroplast, the energy driving that ultimately was from those photons. But in our mitochondria, what we get the high energy from our food, and the food is used to generate a proton gradient, and that is used to make ATP. So this is the final step then. How do we go from that high energy electron that was on an NADP molecule in the chloroplast? How do we go from there to the food that we eat? And the man who figured this out is shown in this picture. His name is Melvin Calvin. He taught biochemistry and chemistry at UC Berkeley for many years. Could even be some people watching this now who may have taken chemistry from Calvin. I know a few professors who did. At the end of World War II, for the first time, biochemists had available in kind of with reasonable access, radioactive isotopes. So Calvin wanted to know, how do plants take carbon dioxide out of the air and use it to make sugar? And so he took radioactive carbon and would feed it to algae that he had growing in these big vats. And the radioactive CO2 would get incorporated into molecules, and he'd stop the reaction as fast as he could to see, where's the first step? Where does that radioactive carbon first get incorporated into some other molecule? And that showed him a series of reactions that has been named after him, called the Calvin cycle. Now, we are not going to go through all of this chemistry here. I want to just hit a few highlights here. So what Calvin found is that his radioactive CO2 ended up on a molecule with 3 carbons in it here, called phosphoglycerate. And so CO2 in the air ended up being this carbon on this 3 carbon molecule. So he thought, there must be some 2 carbon molecule that gets combined with CO2 to make a 3 carbon molecule. Looked and looked and looked and could not find a 2 carbon molecule that was the precursor for this. And the story turned out in the end is that what in fact happened is it would bind to a 5 carbon molecule to make a 6 carbon molecule that immediately fell apart into a pair of 3s. And this is the Calvin cycle. Now, the reason this is a chemical reaction that takes energy. These bonds don't want to form, if you will. So you have to make some highly reactive acceptor molecule. And the way you make that acceptor molecule, this 5-carbon that's going to accept this CO2, is by using the energy that came from those photons. So we said those high energy photons generated an high energy electron, and that eventually ended up on NADPH. Well, the NADPH finally passes that on to these small sugars here, and they're involved in the fixation of CO2 and the formation of sugars. The other thing is, I said that you have to make a molecule here that's a fairly high energy molecule to accept this CO2. And where does that energy come from? In part, it comes from the ATP that was made in the mitochondria and the chloroplast. So this is the connection, if you will, between light energy, photons. An electron comes here, that electron ends up in this molecule, that electron ends up in this molecule combined with additional CO2. And basically what we're doing here is we're synthesizing sugar. This goes on to make table sugar, sucrose. That's carrying this high energy electron that came from the photon. So when we eat plants, we are basically eating molecules that contain the high energy electrons that were obtained from the excitation from photons in sunlight. It's the connection between the kind of solar radiation and then the energy in our bodies. And then we use these sugars to generate proton gradients. And we use those proton gradients in our mitochondria to make ATP. And ATP is what's the energy in our body. Now, this key step here of taking CO2 and converting it into a sugar is done by this very complicated molecule called rubisco. That's because this, the ribulose is the 5-carbon molecule that combines with CO2 to make the pair of 3-carbon molecules. So not only is it a very important enzyme in energy, but it also turns out to be a fairly significant amount of the protein that we eat, especially if you're a vegetarian and you chew on anything that's green, because the plants need so much of this to pull the CO2 out of the air that this is one of the most abundant proteins. This enzyme is the most abundant protein in these green cells. And when we eat the green cells, to us, it's just a source of amino acids. So it goes into our stomach, it gets all unfolded, chopped up into amino acids, and we use it to make our own proteins. All right. So we come here then to a biochemist's summary of life. And that is, for us as animals, life starts with food and oxygen. This is the formula for glucose. So we can take a 6-carbon sugar like glucose and combine it with 6 oxygen molecules. And in doing so, the high energy electrons that ultimately were derived from those excited by photons, those high energy electrons get passed down, losing energy step by step. And as this energy is lost, it is connected to processes such as making ATP. So eventually these electrons end up back on oxygen, and the electron plus a H plus an oxygen gives you water. So we generate the water, regenerate the water. And what's left is the carbon and the CO2. So what plants did was to incorporate CO2 into sugars. We undo. We take them apart back again and regenerate the CO2. In the plants, of course, they can take this CO2, they can combine it with electrons that ultimately came from water to make the sugars, all of this driven by solar power. So when you go out into the woods, go walking in the redwoods the next time, I want you to be properly appreciative of the amazing, sophisticated biochemistry that's going on in the living cells in these trees, and especially in the green tissues. And not only appreciative of the sophistication of the biochemistry, but in the spirit of Thanksgiving, thankful because they are generating the oxygen that we need to breathe, and they are generating ultimately the high energy electrons in the form typically of sugars that we need to drive the action of our bodies. Okay, so with that, I will stop sharing here and open this up to questions. All right, questions from anyone here?
Me again.
Okay, go Mark.
An engineer's question. When you start with the energy that you absorb from the photon, how much of it is available when you get to the sugar at the other end? What fraction?
I think it's when you trying to think here. The number I've seen is about 40%.
40%.
40%.
Wow.
Yeah. And the initial steps, the photochemical steps are really efficient. So when you go from that photon to the first stable form, that NADPH, that's even more efficient than that. Yeah.
But how broad? I mean, okay. Another question is, what fraction of the spectrum are you able to bring into that? How much of the total sunlight is converted into?
That's interesting because it turns out that if you look at where the energy is in the parts that's absorbed. In other words, right. the green is in fact, those photons are less abundant. All right, so it's been optimized to absorb the photons that are most numerous in what's coming in the sunlight.
Oh, okay.
Yeah. All right. Now I realize that was probably the most biochemical lecture we have, but go for it if you have questions. Harriet.
Well, you showed us the image of redwoods, and that would be other plants that would produce the oxygen on land. The end result of the production of oxygen in the ocean, who or what produces that?
Right. Well, that's a very good point because most of the oxygen that we breathe comes from photosynthetic creatures in the ocean, actually. I think it's about 70%. So there's all the phytoplankton, phyto meaning, little plants. And so all of these diatoms and all of these little, some of them are just bacteria, others are slightly more complicated cells. But the bulk of it is these small microscopic little green plants or algae that are in the ocean. They are the major producers of oxygen. And the climate change thing here is they are all pH sensitive. And a consequence of global warming is as CO2 dissolves in the water, it produces carbonic acid. It makes the acid the oceans more acidic. That threatens the life of the molecules that are producing oxygen. It can actually be a quite scary scenario. This is a woman, maybe a few years back, we had Christy Croker, who's in the Ecology Evolutionary Biology department at UCSC. And this is what she's working on is ocean acidification. Lots of people are working on it. Yeah, but ocean is key to the whole thing.
Wow.
Okay. Okay. Any other questions about your energy. Well, let me just say a couple more things about our energy in our bodies. That this is a really surprising amount of ATP that we make. So how long does the ATP in our body last us? And that is if it just stopped for some reason, and I could give you a reason, how long would we last? And the answer is about 45 seconds.
Wow.
So we are constantly making ATP. So if you had a chemical, let's say, stopped protons from moving across your mitochondrial membrane, and one molecule that will do that indirectly is cyanide. So you've heard of cyanide poisoning. Why is it such a nasty, fast reaction? What cyanide does is it blocks the chain of proteins that are pumping protons into your mitochondria, generating that gradient. And once that's stopped, the amount of protons inside those tiny little mitochondria quickly flows out through the ATPase, and they're gone in 45 seconds, and then ATP synthesis stops. And you are using it at a such high rate that what goes first is where do you use all that ATP? The major users in our body are pumps in all of our cells that move sodium from inside the cell to outside the cell, and at the same time move potassium from outside from the bloodstream into the cell. So we're keeping those ions in balance all the time, because the way our nerve cells work is to allow sodium to rush in. You get an electrical pulse, and this pulse will say, move your fingers, or think hard about quantum mechanics, all right? And so that sodium is rushed in, so we've got to pump it back out again. And so those pumps use about 40% of all the ATP, 40% of all the calories from our food are used to drive this synthesis of ATP, to drive the pumps that allow our nerve cells to operate. And it's not just our nerve cells that need those pumps. The way we take in food, the good things, and the way we export waste, the bad things, also depends on these gradients across our membranes. So ATP is the key. Got to have it all the time. And just think about all those little whirring motors in all of your cells. A single mitochondrion probably has about 200,000 of those.
Wow.
A single cell, a liver cell, has about 2,000 mitochondria. And you have 10 to the 13th cells. So the amount of whirring molecular motors in your body is truly an astronomical number. Okay, other question. Now, Lois, and you have to unmute. Oh, we're not hearing you. You're unmuted, but your microphone must be set down or something. Oh, now it went back to mute. Okay, try again. No, not hearing you. Okay.
Write it on a piece of paper and hold it up.
Right, right.
Or put it in the chat.
That too, yeah, okay, all right. So the question here. Okay. All right, any other questions?
I have one more.
Yeah.
I once did a crude estimate of what my muscle efficiency was, riding a bicycle up the hill from the ocean in Carmel to the top of the hill and seeing what my polar watch told me what I burned for calories, and I came up with something like about 20% efficiency for converting calories into motion. I wonder if biochemists have a better estimate of the peak of that than I did.
Yeah, I don't remember that number. We really understand in some detail now how ATP is converted into motion, and it's literally these proteins walking along these cables in our muscle cells. But I don't know that number.
Okay.
But it kind of reminds me of another, energy concept for us to remember. Is questions that Mark's asking about the efficiency. We're saying, 40% are efficient, et cetera. Well, where does the rest of the energy go? And it is almost all goes to heat. And so when we say we use all of this ATP, it's interesting that we generate the heat equivalent of about 100-watt light bulb. And, if you think of a 100-watt incandescent light bulb, you would not want to touch that in your hand. But we are generating that much heat 24 hours a day. And so all of this ATP that we're making is used for bodily functions, but we're far from 100% efficiency. And the part that we're not using to walk or to think or to make molecules goes off as heat. So it's an interesting bit of biochemistry that's exploited this, and that is hibernating animals and newly born babies have special mitochondria in which the protons leak out of the mitochondria. And why in the world would they do that? You lose all that energy. Well, you don't lose the energy. The energy is converted to heat. And so creatures that need to use their food basically to keep your body heat up, like a hibernating bear or a newborn baby, have special what's called brown fat mitochondria that are basically heat generators. So Harriet, you had a question.
Yeah, in that New Yorker article about energy, I think there was some suggestion that different people have different amounts of energy. Is this process something that produces variation in human beings' energy?
Well,
What we call.
Yeah. Well, I didn't see in that, and I would be skeptical if there were differences really at the level of the amount of ATP in the cells of different people. Now, there are people who have rare mutations who have defects in their mitochondria. But boy, if it's at all serious, I think if you had as much as a 3% change in the amount of ATP, it would be nearly lethal. These diseases are so rare because you simply don't survive, past birth if you have any of them. So I suspect that this kind of, when you feel really lethargic, I think this is not directly because your mitochondria are not operating at the same level. I think this is all more hormonal. There's just elaborate system connected to your all of your sensory, what your brain is reading out as to how your body is doing, that's controlled by hormones. And I'm very doubtful that it is actually directly reporting anything that's going on in the mitochondria and your levels of ATP in your body. Yeah, I know, and and now there are so many of these health food supplements, and For you, those of you who've heard me talk about my food and biochemistry course, I'm quite a skeptic about those. Now at Costco, you can get whole pallets of various forms of quinone, Q. So you saw that it formed an important molecule in the chloroplast, and in the mitochondria, it's the same thing. So the idea, if we take a lot of quinone, maybe it will boost our energy levels. And so I was looking this up before the lecture, and the bottom line is, so far the research is just not showing a convincing connection there. So, and as with all of these things, there's rarely just one interaction. So if a molecule does something that you think is favorable, it's very likely doing something else also that you may not necessarily want happening. All right, other questions? Okay, well, I think we can wrap it up for today then. So next week will be Martha Zuniga. She talked to us last year, did a terrific job, and she's going to be talking about immunology and some of the kind of engineering that's being done with immune cells to deal with various medical problems. So Martha's a good teacher, and I hope to see you all there on our first Saturday in December.
Thank you.
Thank you, Barry. I really enjoyed it.
Okay.